guys, here are the list of scientist names from NCERT textbook regarding the NEET biology. The first scientist name is Gregor John Mendel. He is the scientist we see in the principles of inheritance and he is considered as father of genetics. And he conducted experiments in garden pea. Whatever the experiments he conducted, we call it as hybridization experiments. For the first time, he is the one who combined the statistical and mathematical analysis to the biology. And he is the one who has described the Mendel's law. We have three Mendel's law. The first two Mendel's law is from monohybrid cross, law of dominance and law of segregation. Even law of purity of gametes also will comes under law of segregation. And for dihybrid cross, it is law of independent assortment. And next, so the chances or the probability of the gametes and the genotypic formation is being given with a checkerboard that is given by Reginald C. Punnett square. Galton is the one who has given polygenetic inheritance. So we know polygenetic inheritance example is human skin color. And so polygenetic inheritance, human skin color and human height. That is the best example for polygenetic inheritance. Remember, for polygenetic inheritance, one phenotype is controlled by many genes. The reverse will be pleiotropy. One gene will be controlling many phenotypes. Hugo de Veris, Carl Correns. Remember, you remember mutation, then remember Hugo de Veris. Carl Correns. He is the one who has given the incomplete dominance. Von Teschmark. All these together worked independently to rediscover the Mendel's work in 1900. Walter Sutton and Theodore Boveri. Chromosomal theory of inheritance. They studied the Mendel's hybridization experiments together and they made the chromosome, the parallelism of the chromosome and genes. Thomas Hunt Morgan. He is the father of experimental genetics. So he experimentally verified the chromosomal theory of inheritance. And he is the one who has given the term linkage and recombination. He worked on fruit flies, Drosophila melanogaster. Why he suited Drosophila melanogaster is that will complete its life cycle within 2 weeks. And that will give many offsprings. And more observable contrasting traits will be seen. And you can even observe that fruit flies under low power microscope also. And next, Alfred Sturtevant chromosomal maps or linkage maps. Centimorgan is the units he has given for mapping. Henkin, X-body. He studied on insects and he identified that half of the gametes having special body. Half of the gametes are not having special body in male insects. From that he came to know the special body, he named it as X. And the half of the gametes which don't have that X. We see in a grasshopper XO type of sex determination in males. Friedrich Mischer. So he is the one who has first recognized DNA is the acidic material present inside the nucleus. So that's how he termed as nuclein. And he never discussed anything regarding the genetic material, DNA as the genetic material. Next, Maurice Wilkins and Rosalind Franklin, X-ray diffraction data. And based on this X-ray diffraction data only, Watson and Crick proposed the double helical model of DNA and they postulated the semi-conservative method of DNA replication also. They haven't proved, but they postulated. But interestingly, the Nobel Prize is received or shared between three scientists, Watson, Crick and Wilkins. So, but not with Rosalind Franklin because she was died by the time. Erwin Chargraph is denoted by A plus T by G plus C is equal to 1. So, A plus T by G plus C is equal to 1. So, Francis Crick, he is the one who has given the central dogma. Central dogma is none other than DNA to mRNA by transcription, mRNA to proteins by translation, and DNA to DNA by replication. This we call it as central dogma that is given by Francis Crick. and tRNA as the adapter molecule is also proved by Francis Crick only. Frederick Griffith, transforming principle. He worked on Streptococcus pneumonia. So he infected the mice initially with S strain and the mice dead. R strain, the mice was alive. And heat killed S strain, the mice died. Heat killed S and R, infected to the mice and the mice dead. So something is being transferred from heat killed S to R. That is transforming the R to S. So from this, but he couldn't able to say what is transforming, what is transferred and what is making to transform the R to S. And later on, Oswald Ivory, Colin McCarthy. They both determined the biochemical nature of transforming principle, none other than the DNA, but none of them has accepted. They took the transforming principle and they experimented by using enzymes. When they added proteases, the transforming happened. When they added RNAs, the transforming happened. But when they added DNAs, the transforming is stopped. From that they came to know DNA is the genetic material. The first, they are the one who confirmed that DNA is genetic material, but no one accepted by that time. Later on, Alfred Hershey and Martha Chase. These are the two scientists finally chased that DNA is the genetic material by working on bacteriophage. So they taken two batches of bacteriophage. One batch they grown in the radioactive sulfur with S35, and the other batch of bacteriophage they was grown in phosphorus radioactive isotope P32. So in the supernatant fluid of sulfur they absorb the radioactivity. Inside the cell they absorb the phosphorus 32. From that they confirmed DNA is the genetic material. The unequivocal proof of DNA is the genetic material is given by Alfred Hershey and Martha Chase. And Matthew Michelson and Franklin Starr, they experimentally proved that DNA will be replicating semi-conservatively using E. coli, using heavy isotope N15 and light isotope N14. And next, George Gamow, genetic code should be 3 codons. 3 nucleotides. Genetic code should be made up of 3 nucleotides. George Gamow. Hargobind Khurana developed a chemical method of synthesizing RNA molecule with defined base combination. So he synthesized RNA molecule with base combination. Marshall Nirenberg, he is the one put forward the cell-free system of protein synthesis that help deciphering the code. Nirenberg has synthesized the protein. And Severo Oka, so he showed the polynucleotide phosphates also helps in polymerizing the RNA with defined sequence of template independent manner. Francis Jacob and Monod. Jacob and Monod, we remember the Lac-Operon concept. Transitioning regulated system, how the regulated system is seen. Frederick Sanger. Frederick Sanger is the one developed the method of determining amino acid sequence in proteins and developed the principle of automated DNA sequencers, which worked on. Operon and Haldane, they proposed the first form of life could have come from pre-existing non-living organic molecules, RNA, protein, and that the formation of life was preceded by chemical evolution. Louis Pasteur disproved the spontaneous theory, experimentally demonstrated that life comes only from pre-existing life. S.L. Miller experiment on origin of life. He revealed that life originated from non-living components. Charles Darwin, he is the father of evolution, concept of natural selection. Organic evolution comes from interspecific competition. Okay, so Darwin finches, Galapagos Island, and interspecific competition is the one that leads to organic evolution. Adaptive radiation, so and the concept of reproductive fitness, ivanni cheppindi Charles Darwin. Alfred Wallace, he worked on Malay Archipelago, which is group of islands. He have a similar opinion that of Mendel on the concept of reproductive fitness and natural selection. And Lamarck, evolution due to use and disuse organs. Thomas Malthus, he proposed Malthusian theory of population. The theory states that population growth will always tend to outrun the food supply. This theory is thought to influence the Darwin. And next, William Harvey discovered the blood circulation. Hugo de Veris developed mutational theory, saltation. Hardy and Weinberg. So Hardy-Weinberg principle, genetic equilibrium in population. Norman E. Borlaug, he improved the methods of cultivation and developed the robust strain on wheat, semi-dwarf wheat. Alexander Fleming discovered the first antibiotics, penicillium. Ernst Chain and Howard Florey established the full potential of penicillium as antibiotics. Stanley Cohen and Herbert Boyer constructed the first recombinant DNA. Gauss provided the competition exclusion principle. Two closely related species competing for the same natural resources cannot coexist indefinitely, and competitively inferior one will be eliminated eventually. MacArthur, resource partition. So showed that 5 closely related species of warblers living on the same tree were able to avoid competition and coexist due to behavioral difference, like foraging activities. Edward Wilson coined biodiversity to describe the combined diversity at all levels of biological organization. Robert Constanza, he is the one who put the price tag for nature's life support services. Robert May made more conservative and scientifically sound estimated, which places the total global species, which is around 7 million. Alexander von Humboldt observed that region species richness increasing with increased explored area, but only up to certain limit only. David Tillman, his long-term ecosystem experiments states that the plot with more species showed less year-to-year variation in total biomass. So Paul Ehrlich, he provided the revert pauper hypothesis and shows how quality of life is affected with reduction in organism, how the imbalance of ecosystem is created. So that's it. Thank you. These are the scientist name you have to remember for the NEET purpose. Thank you.